All right. I understood that part. You know, while we were trying to search for the strategy, you remember that at a point we had to include signal protein, right?
Yes, sir.
And downstream analysis, we also made mention of exporting protein. All this, we are still trying to identify protein with a peculiar function, of which without the function of such a protein, the parasite might not be able to proliferate or survive within the host. So that was why we had to add the transmembrane domain. Basically, it controls signal transmission and transportation of nutrients. Do you get it? And other soluble molecules that actually helps in energy conversion across different organelles and the cells. So that is why it's important we add the transmembrane domains if your strategy includes signal proteins or exporting protein or the two. Do you get it? You know, there is within different organelles in our body we have cells, cell wall sort of. You know, there's a way they are being stratified. So within the cell, every component of the cell has membrane. Do you get it? An inability of nutrient to cross the barrier, it can lead to the extermination of such a parasite. For instance, okay, let's say for instance in human, most ATP is generated in mitochondria, even in all living cells that are undergoing aerobic respiration. What if the three carbon glucose cannot enter into the Krebs cycle or TCA? I don't know if you understand what I'm trying to say. You know, there is a membrane, there is a barrier. I'm
Yes.
trying to relate this to glycolysis when you have to break down glucose to generate ATP. You know, anaerobically you can break down glucose into pyruvic acid, but pyruvic acid must enter into the Krebs cycle inside that mitochondria to generate more ATP. The same thing happens in parasite. If the membrane we are talking about cannot aid transportation of nutrients or molecules, such such parasites will die of starvation. So the reason why you want to target parasites, target genes that are peculiar to transmembrane domain. That's why we have to add it. Do you get that?
Yes, sir.
Remind you, when you are running strategy that involves signaling and exporting gene, try to add the transmembrane domain. Annabelle, we are waiting for you.
Can you see my screen now?
Not yet.
Is it visible now?
Yes, it is.
Yes.
Okay. So this is so far this is sort of what I've done so far. So it's just an overview of what I've done so far, the results sort of. So I started with identifying in my plant of interest, and from there I went to developing the library and thinking of the strategies to find the protein of interest. So this Triumpheta cordifolia, this is just some background about the plants and how, where it's found, and sort of uses of the plant. So it has economical and medical uses. So it has been used for like even weaving of baskets and stuff like that, and has medicinal uses too. So most reports would tell you that the plant contains alkaloids, steroids, cardiac glycosides, saponins, terpenes, tannins, and flavonoids. They may not go into, what specific ones, but that's what most reports do. And a lot of reports have implicated the plant in, to have antimalarial effect, antibacterial, antifungal, lots of different properties. And one thing of note is that all parts of the plant are pharmacologically useful. So some of the reports I've seen use the flowers, some use the stem, some will tell you it's bark. So all parts of the plant are ecologically useful. So to find proteins of interest, this is just sort of like a note that I just use, looking at what stage, biological activity of that protein and molecular function. And this is the search strategy that I have so far. I'm still working on developing another, but the one that I have so far has given me this protein, which was the one I told you about in the last meeting.
Yeah, let me interrupt you before you proceed.
Okay,
Okay, probably after your presentation, you come back to this strategy and launch it. Let us say. Moreover, on your
okay, I was thinking of doing that now. Okay.
on your search strategy, you can send your link out. You can share your link instead of
Yes,
a
yes.
particular protein. Do you get what I'm trying to say? So you have to go to the entire search strategy, then you can copy your link and probably share with us on our channel. We can also take a look at it. At the same time, you are going to open it here, but you can proceed.
Yes.
No, proceed to your slide before you go to the strategy.
Okay. So basically, the library of phytochemicals that I've been able to get so far from the plants, I put them into a table and then linked it to the PubChem ID and the supporting literature, so I can always know where exactly I got that information from and reference it later. And so this is just that table showing the different compounds. I have about 39 of them. The 40th one is I'm having trouble because of how complex it is. I'm having trouble locating the structure, and I was trying to troubleshoot how I can be able to draw the structure and see if I can put that into a software that can transform it into an SDF file. But that's a question I was going to ask for later.
Yes. Okay. Let me pause you there. You can once you have the name, if you search it and you could not see the structure, probably all you can see is the canonical smile. We can use Chemazon. We can also draw it on Mestro. You can draw it using Chemazon.
Okay.
You can draw it using Marvin Sketch. There are a lot of tools you can
Marvin
use.
Sketch. Okay.
Yes, you can draw.
Okay.
But
I was
meanwhile,
thinking of the Marvin Sketch route, but
yeah, but meanwhile, once you have the name, you can search various databases whether you see the structure. So if you can't see it, then we have to draw it manually.
yeah,
You can
I
proceed.
think that's because these last three, I'll highlight them. These last three compounds were actually novel compounds from this work. So
Okay.
they reported a novel ceramide. That's the second one. A novel this first one and this third one. So I guess that's why I was having trouble tracking this one down. I could get this other two from PubChem. So
Okay.
I'll launch my strategy now. I don't know if I have
Before
to.
you,
Am
before
I still
you,
sharing? Oh,
yes, you are. Before you
okay.
do that,
Yes, sir.
I hope you've harvested all these compounds.
Okay. Let me stop sharing and share
Okay.
in a way that I don't need to keep sharing. Let me just share my
Because,
entire window,
okay.
entire screen. Okay. Can you see my screen now?
Not yet.
Is it visible now?
Not yet. Yes, it's coming up.
Yes,
Okay. Okay. So this is. So I have a folder for it, and I have
Okay.
a folder for the
For your
literature
literature. Okay, that's all
and
right.
for the SDFs.
Okay, yeah, that's all
So
right.
the literature, I also split it out, the ones that specifically gave me where I harvested the phytochemicals from, and the ones
That's
detailing the effects of the compound. So these are the SDF,
okay.
2D SDF. And I remember you said we should download the compound and not the
Yes, not
substances.
structure, not substance. You need
Yes,
to download the compounds.
yes.
Okay, you can move
So
forward.
this is it now. Okay, so for the search strategy, so this is the search strategy. It was actually the second one that I tested that brought me to
Okay.
know. So this is it.
Do you
I
do you
use okay.
let us review your strategy from the very
Okay.
beginning. Which one are you speaking to now? The first or the second one?
This the second one.
The second one. So um let's edit your first step when you have.
Where should I start from the first
Yes, please.
here?
Yes.
Okay.
Go to revise.
Yes. So this was the experiment that I went with, and the reference sample I used the sporocytes, and the comparator sample was delivered at 6 hours after infection.
Oh, what
I
is
was also
your rationale for choosing 6 hours?
I was also going to ask if it was possible that I could
Select everything.
go with all. Like I
Yeah,
tried to select everything and it's
that will not be differential expression. Do you get it?
yeah,
That will not
okay.
be differential. But
Yeah.
probably after this, we quickly run through that. Change your strategy from that 6 hours to 48 hours. Let's see what will happen. Yes.
So just for upregulated, the fold difference was 4 and
Yes.
delta.
Yeah. Run it and see, and then I will tell you. You have more genes compared to the one.
At 6 hours, yes.
Yes.
Okay.
Do you see changes? So what do you want to
So
see?
yes. So now I have 1,585
Yeah. The
compared
reason
to
why you have to select. In Plasmodium life cycle, every 48 hours is the beginning of new cycle, right? As you know.
yes.
So the 6 hour you selected, why you compare that one to your sporozoid? The sporozoid circumvent the cover cell and enter into the hepatocyte, the liver. So the gene upregulated at that particular late sporozoid and the early liver stage are still very similar. Do you get
Okay,
it?
okay.
Compared to when you are
Yes.
having sporozoid and 48
48
hours
8 hours
hepatocyte
post.
stage. Do you get it? So that's why you have to. Meanwhile, when you want to, let's assume you want to look at the 6 hours. That one is more stringent assay that you want to look at. You are dive
Okay.
more into a specific circle. Do you get it? So that's why you have to select at least 48 hours hepatocyte. So let's go to your go term. You can revise.
Okay.
Go down. Or
I
the
use
okay, is that right?
cartridges and
Okay, you can close it. Just close it. Next step is signal peptide.
signal peptide.
Let's go down.
No, I left those ones as is.
No, go up, go up, go up. We use white card, right? Signal peptide. That's all right.
Yes,
We use white
signal
card.
peptide.
Yeah, you can close it. But meanwhile,
Then the
meanwhile, there is another step. Okay. Let's say before that your export.
okay
Okay. No,
before
before.
export.
No, click that signal peptide. Signal peptide. Okay. Go to revise. Cancel it. Cancel it.
I should
Cancel.
cancel it.
Yes. Go to the signal peptide again.
Revise.
No. Insert step before. Insert step before. Okay. Now, under your search box, type signal. Signal. Do you see it?
Predicted or
Yes.
transmembrane domain count.
Predicted signal. Predicted signal. Select that. Select your class. You normally go for the whole thing. That's all right. Run it. Don't worry. Let's see.
The final count has changed.
So initially, your signal peptide was how many?
It was less than this. Now it's the signal peptide. The previous one we just did, or the former.
Do you
This one,
understand what I'm trying? Yes. What was it before?
I don't know the exact value, but it was
Okay.
lower than this.
No. The one you added, remove it. Let me tell
I
you
should
what
remove
you are
this one.
missing. Yes. Just delete. So what do you have your signal peptide?
It's okay. It has been updated.
12.
Signal peptide 6,561.
Now add it again. Then compare your results. No. It's before. No.
Signal peptide.
Yes, before. You have to click signal peptide and add it on before. Signal
It's before.
peptide.
It's after geotemp.
You have to click on that signal peptide you now add before. Do you
Okay.
get it?
I wanted to. Okay.
Yeah. So search for the signal. Let's go for it. No. Only the first one. Oh, jeez.
Sorry, sir.
No, it's all right. You can edit it. Just go back and edit it.
Okay.
Revise. Then I'll check. See what check. Okay, I'm sorry. I run it.
No, this is. Okay.
Let's run it. So, what was the first signal? This is not your result. You are seeing another thing. Seems you edited the wrong
This one.
thing.
I think so. Let me revise. Sorry about that.
I thought as much.
Okay. Yes.
So, what was the first? The first one was about 10, right, or 12?
Yes.
And this one now is 10 and 2.
Yes.
Sorry, hold on.